Everyone, welcome to BioOcean, and this is me Sheethal with you, and today we are here with our new video about immunity, majorly adaptive, otherwise the so-called acquired immunity. And dealing about immunity is one of the introductory part of immunology, so you need to understand the types, the processes, the ways they get administered and all that. So here in this topic we will be focusing on adaptive or acquired immunity. And before getting into the video, if anyone of you are new to my channel, please go and subscribe to my channel and press the bell icon so that you can get the notifications whenever I upload a video. And first of all, we need to understand the term immunity. What actually is immunity? Otherwise, we need to get a definition for immunity. Simply speaking, we can say that immunity is a kind of resistance. But what kind of a resistance, right? So we can define immunity as a resistance exhibited by an individual towards injury caused by microorganisms and their product. Like whenever a person gets injured by any of the injury causing substances, the body will tend to produce some kind of resistance towards that substance. And that resistance provided by the individual towards this injury is called as immunity. And there are several types of immunity. And first of all, we will be starting up with adaptive or the so-called acquired immunity. And here, this kind of immunity, or this is a resistance a person develops during this course of lifetime. And it is mainly of two types, active and passive. Each of them is again divided into two. That is, active immunity is divided into two, natural active immunity and artificial active immunity. Likewise, passive immunity is again divided into two, natural passive immunity as well as artificial passive immunity. So let's start with active immunity. It is a resistance which is developed by an individual as a result of antigenic stimulus, and it involves the active functioning of the person's immune apparatus, leading to the synthesis of antibodies or production of immunologically active cells. Here, what it says that active immunity gets developed whenever a person gets an evoke from an antigen, and towards this antigen, the body, otherwise the immune system of that specific person, will be developing some kind of substances that is called as antibodies. And if you don't know about antibodies, please do click on the I button. I have already created a video on that, and you can watch it there and get the appropriate idea so that you can understand this portion too very well. Okay, so whenever a person gets an antigenic evoke, the person's immune system will be developing some kind of a substance against that antigen, and that is called as an antibody, through which this type of immunity is get developed. Okay, and this active immunity sets only after a latent period which is required for the immunological machinery to be set in motion. And during this development of active immunity, there is often a negative phase, and here at this type of negative phase, the level of measurable immunity may be actually very much lower than it was before the antigenic stimulus. And this is due to the antigen combining with any of the pre-existing antibody and lowering its level in circulation. Once developed, the active immunity is so long-lasting, and the production of antibodies against that antigen is called as an immune response. And as we told earlier, active immunity is of two types, natural and artificial. The former is due to the attack of pathogen, and the latter is by the active immunization through vaccines, otherwise via vaccination. If an individual who has been actively immunized against an antigen experiences the same antigen subsequently, the immune response occurs more quickly than during the first encounter, and this is termed as secondary response. And there is immunological memory conferred by T and B memory cells, and active immunity works by the action of T lymphocytes, which is associated with cellular immunity, and B lymphocytes, which is associated with humoral immunity. Then comes the two different types of active immunity. First of all, starts with natural active immunity, and it results from the infection by a pathogen. We can say it as an example for a person who has recovered from an attack of chickenpox naturally developed a kind of active immunity towards it. But in case of some viral diseases like influenza or common cold, the immunity is so short-lived and it can reoccur in an individual. But don't think that it is not due to the lack of immunizing capacity. But the same clinical picture can be caused by the infection with a larger number of different viruses. The immunity following a bacterial infection is less permanent than following viral infections. In case of syphilis, a special type of immunity called primunation is seen. Here, the immunity to reinfection lasts only as long as original infection remains active, and once the disease is cured, the patient becomes more susceptible to spirochetes pallidum infection again. So what it says here in natural active immunity is that depending on that causative organism or depending on that pathogen, the type of immunity or otherwise the longevity of immunization differs, okay, according to that kind of a specific pathogen. And then comes the artificial active immunity, which is the resistance induced by vaccines. Vaccines is actually the preparation of live or killed microorganisms or their products which is used for immunization. The bacterial products which are used here are called as exotoxins or toxoids. And vaccines are immunological substances that produce specific protection against a particular disease or a particular pathogen. And there are several types of developed vaccines. First of all, it is live vaccine. Live vaccines are actually prepared from live attenuated organisms. So what means attenuation? Attenuation is that to reduce the virulence of an organism, and here this type of viruses are more potent than killed vaccines, and it should not be administered to the pregnant women. In case of second type of vaccine, that is the killed vaccine, killed and attenuated strains of bacteria are used. Sometimes viruses are also used for immunization against diseases, and it gets immunized for diseases like typhoid, smallpox, measles, etc. And the immunity developed by live vaccines is similar to that of the natural infection, though it may be of lower order. The immunity lasts for a year, but booster doses are necessary. Killed vaccines are less immunogenic than live vaccines, and the protection lasts for only a short period of time. So at least 2 doses are required for the production of immunity. The first one is called as the primary dose and the subsequent is called as the booster doses. In killed vaccine, parental administration provides immunity. And that's about vaccines and artificial active immunity. Then comes the second type, which is the passive immunity. Passive immunity is a kind of resistance that is transmitted to an individual in a ready-made form. And here, the recipient's immune system plays no active role and there will be no antigenic stimulus as in the case of active immunity that we saw earlier. But preformed antibodies are administered here. There will be no latent period, but the protection provided is very much effective and immediate after passive immunization. There will be no negative phase, and the immunity here is so transient, lasting normally for days or weeks. There will be no secondary response, and passive immunization is less effective and provides immunity inferior to that produced by active immunization. The main advantage of passive immunity is that it is immediate in action and can be employed when instant immunity is needed. Passive immunity can be natural, otherwise artificial. So start up with natural passive immunity, which is a resistance passively transferred from mother to baby. In human infants, maternal antibodies are transmitted through placenta and through colostrum. Human colostrum is majorly rich in IgA or immunoglobulin A. If you don't know about this type of immunoglobulins and all those, click on the I button. I've already made a video. You can see it there and get an appropriate idea of what it is. And it is only when the baby reaches 3 months that it acquires a satisfactory level of immunological independence. Until then, the maternal antibodies give passive protection. And it is possible to improve the quality of the passive immunity by infants by active immunization of mothers during pregnancy. And then comes the second type, which is last but not the least, that is artificial passive immunity. And this is the resistance passively transferred to the recipient by the administration of antibodies. And here, the agents used are hyperimmune sera of animal or human origin. Sera, or the so-called serum, is a kind of fluid collected from blood and it contains antibodies. One of the example is tetanus immunoglobulins, that is abbreviated as TIG. A special type of immunization is the injection of immunologically competent lymphocytes, and this type of immunization is called as adoptive immunity and it does not have a general application. Instead of whole lymphocytes, here an extract of immunologically competent compounds, otherwise immunologically competent lymphocytes known as the transfer factors, are used. And this has been attempted in the treatment for certain type of diseases like lepromatous leprosy. And so, we understood about passive immunity and the two types, natural passive as well as artificial passive, and along with the adaptive immunity, and also the natural immunity, active immunity too. Active immunity again is divided into natural and passive, and we understood that too. So each of the immunity, that is either active or passive, has two different types, passive, natural as well as artificial. So sorry for the mistake happened, and I hope you find this video helpful, because it is so confusing that the terms get like so much confused, because like more or less similar terms are using in the two different contexts, but the idea, like what is portraying the two different processes is entirely different. Some is due to some antigenic stimulus, while some is due to vaccines, and like that so much difference is there in between these two. I hope you find this video helpful. If you do so, please do like, share and subscribe, and if you have any kind of doubts or suggestions, please let me know through my comment section. And also, if you have anyone by your acquaintances who are interested to learn biology, please do share this video to them too. And thanks for watching. That's all for today. God bless. Thank you.